Hello, welcome to Biology, the scientific study of life. A transcriptional regulation of gene expression during development. As the tissues and organs of an embryo take shape, the cells become visibly different in structure and function. These observable changes are actually the outcome of a cell's developmental history. Extending back to the first mitotic divisions of zygote. However, the earliest changes that set a cell on a path to specialization are subtle ones, showing up only at the molecular level before biologists know much about the molecular changes occurring in embryo. They coined the term determination to refer to the events that lead to the observable differentiation of a cell. At the end of this process, an embryonic cell is irreversibly committed to its final fate, and it is said to be determined. If a committed cell is experimentally placed in another location in the embryo, it will still differentiate into the cell type that is its normal fate. Today we understand determination in its terms of molecular exchange, molecular changes. The outcome of determination observable cell differentiation is marked by the expression of genes of tissue specific proteins. These proteins are found only in a specific cell type and give the cell its characteristic structure and function. The first evidence of differentiation is the appearance of mRNA. As for these proteins, eventually differentiation is observable with a microscope as changes in cellular structure. In most cases, the pattern of gene expression in a differentiated cell is controlled at the level of transcription. Differentiated cells are specialists at making tissue-specific proteins. For example, as the result of transcriptional regulation, liver cells specialize in making albumin, and lens cells specialize in making crystallins. Indeed, lens cells devote 80% of their capacity of protein synthesis to making crystalline proteins, which enable the lens to transmit and focus light. Skeletal muscle cells are another instruction example. The cells of skeletal muscle are long fibers containing many nuclei within a single plasma membrane. They contain high concentrations of muscle-specific versions of contractile proteins, myosin and actin, as well as membrane receptor proteins that detect signals from nerve cells. Muscle cells develop from embryonic precursor cells that have the potential to develop into a number of alternative cell types, including cartilage cells of fat cells, but particular conditions commit them to becoming muscle cells. Although the committed cells appear unchanged under the microscope, determination has occurred and they are now myoblasts. Eventually, myoblasts start to churn out large amounts of muscle-specific proteins and fuse to form mature, elongated, multinucleate skeletal muscle cells. Notice this figure. Notice here nucleus embryonic precursor cell. This one present closer look. Notice that. Master control gene myo-D DNA. Notice this one. Closer look to all the stage. One determination myoblast determined. Notice also closer look. mRNA. Notice myo-D protein transcription factor. Determination signals from other cells lead to activation of master regulatory gene called myo-D, and the cell makes myo-D protein, a transcription factor. The cell, now called a myoblast, is irreversibly committed to becoming a skeletal muscle cell. Notice that stage 2 differentiation. Closer look. Notice that Um, MUD protein stimulates the MUD gene further and activates gene encoding other muscle-specific transcription factors, which in turn activate genes for muscle proteins. MUD also turns on genes that block the cell cycle, thus stopping cell division. The non-dividing myoblasts fused to become mature multinucleate muscle cells, also called muscle fibers. Notice that mRNA, MUD, and notice also mRNA, another transcription factor. Notice also mRNA used in other muscle proteins and cell cycle blocking proteins. Okay, let's okay, researchers have worked out what happens at the molecular level during muscle cell determination by growing myoblasts in culture and applying some of the technique. They first produced a cDNA library containing all the genes that are expressed in cultured myoblasts. The researchers then inserted each of the cloned genes into a separate embryonic precursor cell and looking for differentiation into myoblasts and muscle cells. In this way, they identified several so-called master regulatory genes whose protein products commit the cells to becoming skeletal muscle. Thus, in the case of muscle cells, the molecular basis of determination is the expression of one or more of these master regulatory genes. To understand more about how commitments occurs in muscle cell differentiation, let's focus on the master regulatory gene called MUD. This gene encodes MUD protein, a transcription factor that binds to specific control elements in the enhancer of various target genes and stimulates their expression. Some target genes for MUD encode still other muscle-specific transcription factors. MUD also stimulates expression of the MUD gene itself, thus perpetuating its effect in maintaining the cell's differentiated state. Presumably, all the genes activated by MUD have enhancers recognized by MUD and are thus coordinately controlled. Finally, the secondary transcription the secondary transcription factors activate the genes for proteins such as myosin and actin that confer the unique properties of skeletal muscle cells. The MUD protein is powerful. Researchers have been able to use it to change some kinds of fully differentiated non-muscle cells, such as fat cells and liver cells, into muscle cells. Why doesn't it work on all kinds of cells? One likely explanation is that activation of the muscle-specific genes is not solely dependent on MUD, but requires a particular combination of regulatory proteins, some of which are lacking in cells that do not respond to MUD. The determination and differentiation of other kinds of tissues may play out in a similar fashion. Cytoplasmic determinants and cell signals in cell differentiation. Explaining the role of MUD in muscle cell differentiation is a long way from explaining the development of an organism. The MUD story immediately raises the question of what triggers the expression of that gene, and then raises a series of similar questions leading back to the zygote. What generates the first differences that arise among the cells in any embryo, and what controls morphogenesis and the differentiation of all the different cell types as development proceeds. As we saw in the case of muscle cells, the question comes down to which genes are transcribed in the cells of a developing organism. Two sources of information used to varying extents in different species tell a cell which genes to express at any given time during embryonic development. One important source of information early in development is the egg cell's cytoplasm, which contains both RNA and protein molecules encoded by the mother's DNA. The cytoplasm of an unfertilized egg cell is not homogeneous. Messenger RNA proteins, other substances, and organelles are distributed unevenly in the infertilized egg, and this heterogeneity has a profound impact on the development of the future embryo in many species. Maternal substances in the egg that influence the course of early development are called cytoplasmic determinants. After fertilization, early mitotic divisions distribute the zygote cytoplasm into separate cells. The nuclei of many of these cells are thus exposed to different cytoplasmic determinants depending on which portions of the zygotic cytoplasmic a cell received. Notice this figure. The set of cytoplasmic determinants in a particular cell receives helps determine its developmental fate by regulating expression of the cell's genes during the course of cell differentiation. The other important source of developmental information, which becomes increasingly important as the number of embryonic cells increases, is the environment around a particular cell. Most important are the signals impinging on an embryonic cell from other embryonic cells in the vicinity. In animals, these include contact with cell surface molecules on neighboring cells and the binding of growth factors secreted by neighboring cells. In plants, neighboring cells. In plants, the cell-cell junctions known as plasmodesmata can allow signal molecules to pass from one cell to another. The molecules conveying these signals are proteins expressed by the embryo's 1 genes. The signal molecules cause changes in nearby target cells, a process called induction. In general, the signal molecules ascend a cell down a specific developmental path by causing a change in its gene expression that results in observable cellular changes. Those interactions between embryonic cells eventually induce a differentiation of the many specialized cell types making up a new organism. We will learn more about cytoplasmic determinants and induction in the next section, where we take a closer look at some important genetic and cellular mechanisms of development in the 3 model organisms: Drosophila, C. elegans, and Arabidopsis.